you so much for having me. We'll state off. So I'll just start off with my disclaimers and my acknowledgements. I really want to thank the organizers, especially Thermo Fisher, for inviting me for the talk. So being a clinician, I will just want to start with a case. So this was a newborn who was born to a healthy, non-consanguineous young couple in Jaipur as their first baby, who did well for a few days, but then started getting lethargic and with poor oral intake, and he started going, in encephalopathy, and so he required admission with the usual testing and the treatments, thinking about this to be some sepsis, some infections. They continued the treatment, but there were some abnormal things on the usual testing. So like the blood counts were deranged, the liver, the kidney functions, they were all deranged. So something was amiss, and even the blood cultures were all right. So metabolic testing was then taken on, and there were a lot of metabolic derangements in the baby, which led to the suspicion of an inherited metabolic disorder in the baby. So some metabolic tests were done, and they did include the mass spectrometry, and that's the reason why I put in this case. So of course the child was referred to our hospital and was managed and successfully. So the metabolic testing that I'm alluding to was mass spectrometry. So it is a TMS, tandem mass spectrometry, which is done on a dried blood spot, and along with a urine sample, which goes for a gas chromatography mass spectrometry, and that both of them are required to get to a genetic diagnosis of a metabolic disorder. So the GCMS in this baby showed huge peaks of methylmalonic acid, and which gave us the diagnosis of methylmalonic acidemia. So another case came a few months later. This was an infant, not a neonate. So this baby was born to first cousins, a third degree consanguineous couple, and had presented earlier with respiratory problems, frequent respiratory problems, which had not been identified to the depth. But when the baby came to us, the main thing was that the baby had acidosis and there was failure to thrive. And there were a few other pointers, like the urine ketones were positive. So that led us to investigate the child for a further metabolic disorder. So again, the same tests were sent, the TMS on the blood as well as the urine sample. Both of them were corroborating with the diagnosis of methylmalonic acidemia. So basically, this is a genetic metabolic disorder which is eminently treatable. So both of these children have been on treatment and are doing very well. They are now a few years of age now. So what is metabolic disorder? So this slide just shows a complete complication of all the metabolic interlinked disorders, pathways, but I'm not going to touch into everything. I just want to tell you about a little bit about metabolism so that we can get to our applications. So metabolism, of course, is the sum total of all the chemical reactions that keep happening in our body. And inborn errors of metabolism arise due to disruptions in any of these metabolic pathways. So mainly there are 2 or 3 mechanisms where the disorders arise. The first thing is a deficiency, because the metabolic pathway leads to production of some important metabolite which may be deficient and then give rise to problems, or it could be due to accumulation of the metabolites which have now stopped because of the block, and of course also due to the transport. So the laboratory testing. So methylmalonic acidemia was a small molecule disorder, which means the molecules that are in the pathway are small in size. They can cross the blood-brain barriers as well as the cell membranes and cause toxicity. So laboratory diagnosis depends upon looking at these metabolites, and while in a traditional way we are only able to do a few chemical tests like ammonia, lactate, and so on, it is the technological advancement that has really helped us. So these analytical methods that have now been employed really help us in getting to a confirmatory diagnosis. So these are very many, like tandem mass spectrometry, GCMS, LC-MS-MS, HPLC, and other electrophoretic techniques. So I come to mass spectrometry now because that's the main reason why I have been here, to tell you about its applications for diagnostics in children, especially in inborn errors. So mass spectrometer is, this is a method which employs two mass spectrometers in tandem with each other. A sample is sent, into an ionization chamber, where the ions are separated, and then they are made to enter into two chambers, one after the other, so that they are separated according to the mass to charge ratio, and ultimately they get detected, and the computer analyzes it in terms of signals, and there are peaks that we can identify and get an idea of about the metabolites, the quality as well as the quantity. So this helps us in making a diagnosis of a particular disorder. So the story of mass spectrometry is not young. It started more than a century ago, and there have been along the line many Nobel Prize winners because of this. So I would just say that it really took off in the 80s when it was developed for diagnostics, and then it was mastered in the 90s, and of course the application started in the 90s. And for me, the real impact or the real application for diagnostic in inborn errors in India started in the noughties, like year 2000 to 2010. Many laboratories have now come up in India who are doing the diagnostics in this way. So the main test that we employ, the MSMS, is able to give us the answers for acyl carnitines, the amino acids, as well as organic acids. The newborn screening is one of the biggest success stories in the world, and mass spectrometry has a lot to do with it. Although it was started in 1960s as a one test, one disorder detection, but it was the mass spectrometry which actually changed the game. So back in the late 90s, when it was started in the newborn screening area, the newborn screening went from one test, one disease, to one test, multiple diseases for a diagnosis. So Australia was one of the first countries that employed this use of tandem mass in newborn screening and called it expanded newborn screening. So I've had the opportunity to be in Australia at that time when I was training, and we brought out a very eminent paper which gave us the impact of this technology in saving lives. So here was a disorder, glutaric aciduria type 1, which can only be detected by tools like these. And when you study this in a newborn who has not yet had any symptoms, and you start the treatment, we get excellent results. So up to 90% of the children who are screened at the newborn stage for this disorder are surviving and doing well, as opposed to the pre-newborn screening era, or maybe a situation here in India where we don't have newborn screening for this, is that only 10%, or maybe I can put it this way, 90% of the children with glutaric aciduria will either die or face severe morbidity. So such is an impact of this mass spectrometer. So inborn errors are a group of disorders that are expanding exponentially. In 2019, there is an international classification of inherited metabolic disorders, which has now come up to like 1,400 or 1,500 disorders. So the technology is assisting in all the groups of disorders in detection as well as in monitoring, and so that it is also helping the treatment. So just to simplify the classification of inborn errors, there are small molecule disorders like the ones that I told you at the beginning. Then there are complex molecule disorders such as the storage disorder, the lysosomal storage disorder are a common group. And of course, the energy deficiency disorders like the mitochondrial disorders and the fatty acid oxidation. So every group has so there is a role of mass spectrometry in diagnosing majority of the disorders in small molecule and a few of them in the complex molecule and energy deficiency as well. So the number of applications of this keeps on increasing. This is just a snapshot of one test which can produce which can detect so many metabolic disorders, the GCMS. But this can also be utilized in other body samples like the blood, CSF. There are so many applications of LC-MS now, the liquid chromatography tagged in along with the mass spectrometer, which increases its sensitivity and which can go beyond the scope of a few genetic disorders, metabolic disorders, let's say the neurotransmitters, even the complex molecules like biomarkers for lysosomal storage disorders. So all these disorders can be diagnosed by this technology. So this is I borrowed this slide from Mr. Bharuja from Thermo Fisher. So just to tell you the application of this mass spectrometer, I am here basically in just this small biochemical genetics part where I have told you about the application in metabolic disorders, but going beyond, there is a lot of role of mass spectrometer in detecting the endocrine disorders, like all the hormones, the vitamin levels, the peptides, even in therapeutic drug monitoring. I'm sure the panel that comes after this will be discussing this whole aspect. And not to forget the toxicology aspect. It's really, really clinically important that it has a big role in picking up the toxic metabolites, whether they are inherited or just acquired, or there's a lot of role in forensics for picking up the toxicology. So now we move beyond the targeted mass spectrometry into the untargeted territory, like my previous speaker had said. So because we know just a few of these disorders that can be picked up, but what if we expand this technology to try to understand what's happening even beyond that? So that's the idea of going into the next generation metabolic screening, which is the untargeted metabolomics, which is also happening in the field of inborn errors of metabolism. So here we go from known to unknown, and we can catch more than 10,000 metabolites in a single sample. And of course, the way the test is being used is to make use of the technology. There is also annotation which uses the human metabolome database to identify certain peaks and metabolites. So how is it useful for us as clinicians? Basically, there is, at this present time, a lot of genomics is being done where DNA is being analyzed, the genetic variants are coming out, but we are still lagging behind the understanding of these variants. So there is a condition called variants of uncertain significance. And when we come across such a situation where in a genomic test you have identified a variant where you cannot understand the significance, this test can come in handy. So it can help, sort of replace or akin to a functional or biochemical substitute to try and understand the diagnosis of a particular individual. So I would just like to conclude here by saying that, of course, diagnostics is super expanding in the field of IEMs, and of course, it is boosted by the technologies such as mass spectrometry. So the newer assays are not only just sensitive, but also very accurate, and they need less time, and the cost is coming down as we are applying it more and more. So the ultimate aim as a clinician, as a doctor, is to treat our patients, and the earlier we detect such disorders and more accurately we detect, the best we can manage and save lives of these individuals. Thank you so much.